Chapter 8 Linkage, Crossing Over, Recombination, and Eukaryotes. Linkage. Genes sort independently if they are on different chromosomes but show linkage if they are on the same chromosome. Independent assortment. Two genes on two different homologous pairs chromosomes. Big A, big A, big B, big B. Small a, small a, small b, small b. When they become gametes, you have big A, big B, big A, small b, small a, big B, and small a, small b. But in linkage, two genes with single pair homologs, no exchange occurs. And in your complete linkage, only parental non-crossover gametes are produced, meaning that they will be exactly same as the parents. Linkage. Two genes on single pair homologs, exchange occurs between two non-cis chromatids. Then you have the crossover gamete and the non-crossover gamete. If crossing over between two linked genes occur between two non-cis chromatids, both parental non-crossover and recombinant crossover gametes are produced. So how far? The degree of crossing over between any two loci on a single chromosome is proportional to the distance between them, known as the interlocus distance. In the picture, you can see locus for the first gene containing allele big P or small p, and then the other is big Y, small y. If a complete linkage exists between two genes because of their close proximity and organisms heterozygous at both loci are mated, a unique F2 phenotype ratio designated the linkage ratio results. Genes on the same chromosome are part of a linkage group. The number of linkage groups should correspond to the haploid number of chromosomes, because if you have 23, then you have 23 linkage groups. The percentage of offspring resulting from recombinant genes depends on the distance between the two genes on the chromosome. Crossover, it is a phenomenon where the chromosomes, which are synapses, they wrap around each other to create chiasmata. They are points of genetic exchange. During crossover, synapse chromosomes in meiosis wrap around each other to create chiasmata, which are points of genetic exchange. Two genes located relatively close to each other along chromosomes are less likely to have chiasma to form between them, and it's likely that crossing over will occur. RC frequency. The recombinant frequency between linked genes are additive, and the frequency of exchange is an estimate of relative distance between two genes along the chromosome. Map units. One map unit is defined as 1% recombination between two genes on a chromosome. Map units are often called centimorgans and are relative distance, not exact distances. A single crossover SCO alters linkage between two genes only if crossover occurs between these two genes. Picture A, you can see exchange between the non-sister chromatids, big A, big B with small a, small b. In gametes, you will have a mixture. Big A, big B, big A, big B, small a, small b, small b, small b. So between them, there is no crossover happening. So there is unchanged alleles between A and B. But if the crossover happens between A and B, then you might have A and B in a mixture. Big A, big B, big A, small b, small a, big B. Because in between A and B, there is a crossover. Here you can see, you can have 4 types of gametes, 2 non-crossovers, big A, big B, small a, small b, and 2 crossover gametes, big A, small b, and small a, big B. Single crossovers can be used to determine the distance between two linked genes, but double crossovers DCOs can be used to determine the order of 3 genes on the chromosome and the distance between the linked genes. To study double exchanges, 3 pairs of genes must be investigated, each heterozygous for 2 alleles. The expression frequency for double crossover is much lower than that of a single crossover gamete class. So this is what's called triple mapping. In triple mapping, parent must be heterozygous for all 3 genes under consideration. It is cross-constructed, so genotypes can be determined by offspring phenotypes. Many offspring produced to get representative sample of crossover events. So the 3-point mapping is shown on figure 5.8. So this is the figure 5.8. So you have a parent which is YWEC, YWEC, and then the other, the Y plus, W plus, EC plus, which is wild. So the parent will be crossed together. Then you have gametes which are YWEC, Y plus, W plus, EC plus, and the Y chromosome. Then you will have this parent which is heterozygous, Y, Y plus, W, W plus, EC, EC plus, with a parent which is YWEC and Y, when that is a male. So, the non-crossover F2 phenotypes occur in the greatest proportion of offspring. Double crossover phenotypes occur in the smallest proportion. Because the F2 phenotypes complement each other, one is wild type and one is mutant for all three genes, they are called reciprocal classes. Meaning that if you look at the F2 phenotypes, you can see 3 alleles, they will be complement to each other. You can see one offspring is just Y, but the other is W and EC. Then you can see an offspring which is EC only, but the other is Y and W. Some of them even have two, YW and the other EC. So they're actually a crossover that happens that causes them to be either one allele and then the other will have their remaining two alleles. So that was called reciprocal. So how do you calculate the distance between the alleles? The distance between two genes in the triple cross is equal to the percentage of all exchanges between them and includes all single and double crossover. So if you can see this allele, big A, big B, big C, big A, big B, big C, if parental, meaning that there is no crossover. But if there is a offspring which is different slightly from the parent, as you can see in the second one, small a, big B, big C, meaning that between A and B there is an exchange. So it is single crossover gamete. But what the third one? Big A, big B, small c. Small a, small b, big C. Where does the crossover happen? Crossover happens between B and C. What the one that is the last one? Small a, big B, small c. Big A, small b, big C. Meaning that the crossover happens twice between A and B and between B and C. So determining gene sequence, you assume that the sequence is YWEC, but we do not know whether it is the correct order. So in theoretical sequences, you have YWEC. Okay, for the double crossover gamete will be Y in the middle. So what's left will be, you will see the mutation for Y, which is yellow. So number 2 will be EC in the middle. If EC in the middle, means that if double crossover happens, you can only see the EC allele as a phenotype, which is achinous. And number 3, what if W is in the middle? In double crossover, W in the middle, so you will see the mutation white. That's the mutation that is the lowest. Okay, so you have method 1 and method 2. In method 1, you look at the reciprocal non-crossover phenotypes and test each one of the 3 possible orders to see which one gives you the observed double crossover phenotypes. One which does that is the winner. Method 2, you look at the non-crossover phenotypes, but you look at the single allele that has been switched. This allele will be by itself, alone. So it is no longer associated with the mutant alleles. Meaning that in double crossover, you know A, B, C, A and C has moved to the other chromosome. What's left will be B. So that one by itself, it is the one in the middle. So let's try an example with maize. In maize, you have 3 linked genes: BM, bromidrib, V, virescent seedling, and P, purple aileron. So you have some possible allele arrangements. Is it V in the middle? Is it PR in the middle? Or is it BM in the middle? So all of these are the possible arrangements. So let's see which one is correct. So this is the results of the cross. You can see the plus, VBM, PR, plus, plus. These are the non-crossover. Okay, because they are the parent. And then you have plus plus BM, PR, V plus. It is a single crossover. Meaning that between these two parents, one allele has jumped ship to another chromosome. Then you have plus V plus PR plus BM. So this also is a single crossover, meaning that one has jumped ship, the other remain. Then what about the other one? PRVBM+++, means that 2 has jump ship, leaving only 1 behind. So that's what it's called the double crossover. So take note of the total number and percentage. Which one is the biggest? Which one is the smallest? So as a rule of thumb, the biggest number is the non-crossover, and the smallest number is the double crossover. If you look at A, it is plus VBM, V in the middle. So in this arrangement, the one that you might see as the double crossover would be V. But if you look at the table, what is the one that's left behind? Okay, so you can see plus VBM, PR++. These are the original. Okay, so which one has moved to the other side? It's just PR. The V and BM, they remain. So meaning that the actual middle is the one it is PR. Okay, so that's the double crossover situation. V, PR, BM. So the final map is 22.3 between V and PR and 43.4 between PR and VM. So the expected frequency of multiple exchanges we can predict. We predict using big C. Capital C is the number which is observed over expected. Difference will be the number if you minus 1 from the C, meaning that it will be the one that reduces the number of multiple crossovers. If the interference is positive, you will have fewer double crossover. But if it is negative, meaning that you have even more double crossovers. When the two genes are close together, the accuracy of mapping is high. As the distance between them increases, the accuracy of mapping decreases, so you have positive interference. So here, you can see two exchanges between linked genes which are far apart on a chromosome. It involves 2, 3 or all 4 strands. It can result in production of different percentages of recombinant chromatids. So theoretical will be like that in the red line, but actual much lower than it is theoretical. So Drosophila genes have been extensively mapped, as we have learned from the previous chapter. These are all of the mapped segments in Drosophila chromosome. So in Drosophila chromosome, all of these actually control one trait within Drosophila species. If they are mutated, then the trait will not be shown. So what is lot score? Lot score is the logarithm of odds score, which is used for genetic linkage analysis. So lot score, we compare the probability of obtaining the test data if two loci are linked versus obtaining the data if the two loci are not linked. So it relies on probability calculations to demonstrate linkage between two genes and organisms, in which linkage analysis relies primarily on pedigrees. So this is the human chromosome. We have hybrid cell lines. All of these are mapped portions of the chromosome. So you have gene product expressed, plus, minus. So chromosome mapping is not possible with DNA markers and annotated computer database. So markers represent landmarks along chromosome. We use RFLP, microsatellites, and SMPs. The recombination occurs between mitotic chromosomes. In Drosophila, in certain fungi, homologs pair during mitosis, allowing crossing over to take place. So serial exchange occur during mitosis but do not produce new allelic combinations because serial chromatids are actually the same, clones of each other. The end.